Hi, everyone. I'm Melanie Goldie, the CEO of Tally Health. Thank you so much for joining us today for our latest members-only webinar. We've heard a lot of interest in our community about wanting to know how other people are doing with their TallyAge. We did a deep dive to understand this in more detail, and really looking at the first 10 months of Tally being out in the world and what impact we've had on our membership community at large. Today, we're going to share with you what we've learned in terms of impact to our membership, and have a discussion centered around best advice for lowering your TallyAge. With me today, some of you may know Dr. Adiv Johnson, who is our Director of Research and Innovation at Tally Health. Adiv completed his bachelor's degree in molecular and cellular biology and his PhD in physiological sciences at the University of Arizona. After successfully defending his PhD, he went on to complete a postdoctoral research fellowship in the Department of Ophthalmology at Mayo Clinic in Minnesota. Adiv has been engaged in aging research since 2008, and to date has published 38 publications in scientific peer-reviewed journals, including prestigious ones like Proceedings of the National Academy of Sciences, Aging Cell, and Aging Research Reviews. His first publication, quite interesting, looked at the effects of caloric restriction in mosquitoes, and his most cited work is a 2012 paper exploring the relationship between DNA methylation, aging, and age-related disease. We're going to start with a quick presentation from Adiv, and then we'll go right into live Q&A from you. So please do drop your questions in the chat box below at any time. We can collect those even during that presentation. Without further ado, Adiv, over to you.
Awesome. Thanks so much, Melanie. I appreciate that very kind and generous introduction. And I'm very grateful to everyone that's tuning in today. There's a lot of things you could do with your time, and you chose to be here, which is fantastic. So let me just get this slideshow going. I have a fairly brief presentation because we want to maximize time for live questions. And I will be talking about the TallyAge test and some really exciting learnings and insights that we've gained. And I'm just going to briefly go into 3 different sections during this webinar portion. First, this might be a refresher for many. I'll talk about the TallyAge test and what makes it so unique and innovative, including why it's a next generation test and why that matters. I'm going to talk about the holistic lifestyle associations that we see for TallyAge and share the top 5 factors that most significantly correlate with the results. And I'll end with what I think is the most exciting part of this, some very interesting user trends we've seen for both the lifestyle survey and the TallyAge test over the last year. And before I get into the specifics of the TallyAge test and what makes it so unique and innovative, I just want to introduce the concept of an aging biomarker. This is a very basic concept in the field of biogerontology. Unfortunately, virtually everything changes with age. Our gene expression patterns become dysregulated, the methylation landscape becomes aberrant, protein expression radically changes, cells lose telomeres, they become senescent, muscles diminish with age, brain volume shrinks, skin wrinkles, et cetera, et cetera, et cetera. But there's a very positive side of all this, which is some of these changes are very predictive of outcomes. And these changes that are predictive, that are providing insights into how we're aging, are known as aging biomarkers. And I'll give you a really classical example here. Grip strength is essentially a measure of how well your muscular and skeletal systems are performing. And you can measure it very simply with a device called a dynamometer or by hanging from a bar and seeing how long you can hang. It's actually quite challenging to do much more than a minute or 2. If we were to take a group of older adults, say 65 plus, and measure their grip strength and follow them for 20 years, we would almost certainly find that people with a high grip strength were much more likely to be alive by the end of the study period. And not only that, much more likely to be functional and healthy during follow-up. Thus, it's an aging biomarker. And there are many others out there, including newer ones like epigenetic aging clocks. So an epigenetic aging clock is a more contemporary biomarker of aging. The first ones were published in 2013. And essentially, they use machine learning and DNA methylation information to produce an age output that is referred to as DNA methylation age or epigenetic age. As a quick reminder for what methylation is, we have about 3 billion base pairs in our genome, and about 30 million of those are eligible to receive a chemical methyl group, which is a carbon atom with 3 hydrogen atoms attached to it. And our cells use methylation as a really powerful tool to either hide or make visible DNA. And this affects gene expression, protein expression, and cellular function. And indeed, epigenetic alterations are a well-established primary hallmark of aging. And when we look at a cohort of people, methylation patterns tend to change in very predictable ways. Some parts of the genome gain methylation, others lose methylation. And we can use this information and these trends in conjunction with machine learning to estimate how old somebody is epigenetically. Now, an important distinction here, there are 2 types of epigenetic aging clocks. First generation clocks, while innovative and useful, mostly use CPGs that associate with age. And this is quite interesting from a forensics perspective, but it's less interesting from an aging biomarker perspective. And that's why we care more about next generation epigenetic aging clocks that utilize CPGs, DNA methylation sites, that not only change with age, but relate to health as well. And generally speaking, if you compare next generation clocks to first generation clocks, what you find is that next generation clocks are much more predictive of health outcomes, including mortality and morbidity. So it's quite interesting to us. So what makes TallyAge a next generation test and such a unique test? Well, first of all, we collected data from a really impressively large, diverse cohort, more than 8,000 people spanning an age range of 18 to 100 years. This is almost the full age range of adults. We tragically didn't get up to 122, which is the current record for lifespan, but we got the majority of the lifespan that's out there. We had really excellent gender parity, and the data set was ethnically and racially diverse. And this all matters a huge amount because we want this test to be generalizable to as many types of people as possible, and we want it to work well. The second thing we did that's really critical is we optimized this clock for test-retest reliability. Specifically, we asked 190 people to send us 2 samples within a 24-hour period. And we looked at which DNA methylation sites were more stable or more noisy during that time frame, and we filtered out the noisy sites. And the result of this is we were able to maximize signal and minimize noise, which is critical for an aging biomarker. And lastly, this is the part that makes it especially interesting from a next generation perspective. We trained the model to associate with lifestyle factors that are known to maximize longevity. For example, exercise and sleep. And the specific list of lifestyle factors can be found here. Specifically, we saw that TallyAge significantly correlated with how much of a diet is plant-based. And that matters because we know that a Mediterranean diet, which is a plant-rich diet, is something that is pro-longevity. We also see that vegetarians and vegans tend to live longer than their omnivore and carnivore counterparts in the United States. TallyAge also significantly correlated with weekly exercise, with perceived stress, sleep quality, social satisfaction, smoking status, and tragically, alcohol intake. And I want to note here that all of these associations were significant after we controlled for many different factors. And many of them were extremely, extremely significant. So, which of these are the most substantial in terms of their association with TallyAge? Well, I didn't list it in the previous slide, but it is technically a lifestyle factor. We saw that body mass index, BMI, which is measured given inputs of height and weight, and is a rough proxy for overall energy balance, was the most significant factor that correlated with TallyAge. Now, BMI, I have to add, is a great population level biomarker. On an individual level, it's less optimal because of so many things that factor into it, like bone density and muscle mass. But it's really interesting nonetheless, given that BMI is quite predictive for health outcomes on a population level. I don't think anybody will be surprised to see that smoking status was the second most significant factor. Unfortunately, smoking is something that accelerates aging. It radically increases cancer risk of vision loss, and early death. So it is both expected and also a sanity check that the model is performing the way we want it to. Next up, this is sad news, is weekly alcohol intake. We found that any amount of alcohol increased TallyAge, and that the only average TallyAges that were negative were in people that didn't drink alcohol at all or only had 1 to 3 drinks per week. And some of you may be aware of this, but there's really a growing body of evidence suggesting that the safest amount of alcohol to drink is either none or very little. So this finding is very much in line with more recent higher quality research that's been coming out. The next two, I'll admit, were quite surprising to me. I would have expected to see exercise or sleep quality or something else on the list. Instead, we found social satisfaction and stress. And there's a growing body of literature and evidence essentially arguing that creating a divide between mental health and physical health in some ways is arbitrary. We know that mental health can profoundly affect physical health and vice versa. So in the context of that research and that knowledge, it is really quite intriguing to see two more mental health type factors among the top 5 that are the most significant. So as an example of how this distribution looks, I really want to hammer home the alcohol factor again. We have a graph here with delta age on the y-axis, which is the differential between TallyAge and chronological age, ranging from -10 to +10, and most people are within a few years of the chronological age. And on the x-axis, we have weekly alcohol intake, ranging from 15 or more drinks a week to no drinks a week. And the number next to each of those numbers, each of the weekly intakes on the x-axis, is the average TallyAge. And what you see is that with every reduction in intake, TallyAge continues to go down, with the difference between no drinks and 15 or more drinks being about 2.7, nearly 3 years. So again, sorry to be the bearer of bad news, but it's not looking great for alcohol. Now, I want to talk about how our lifestyle survey and TallyAge test have changed over time. And we launched last year in February and collected quite a bit of data points. We had people take the survey multiple times. And what we found was astounding, which is that 74.8% of users improved their lifestyle survey. And the average gain in points was 5.2. And for those that improved, they improved by 8 points total. Now, this is incredibly non-trivial. To give you an example of how impactful this is, a gain of 5 points would be equivalent to somebody going from not doing any strength training to doing strength training multiple times a week and getting more sleep every night. And we know that those changes on their own would substantially shift someone's aging trajectory and really increase the likelihood of having a longer health span. So this is really exciting and very impactful. Likewise, we've seen that over the course of last year, where we collected 3 different time points in under 12 months, and by the way, we also mailed out more than 20,000 kits by the end of last year, we saw that 62.4% of users lowered their TallyAge. And the average reduction was minus 0.73 years. And for those that lowered, the average was minus 2.34 years. So I want to focus in on this central number here of minus 0.73. In telehealth's first paper that we published in Aging Cell called Human Age Reversal, Fact or Fiction, we created tables of clinical trials that used aging clocks before and after an intervention. And if you look at studies that had an intervention like a plant-based diet and exercise or a Mediterranean diet or something else over the course of 1 to 2 years, you find that the typical degree of change was in line with what we see here, which is minus 0.73 years. So it's really exciting to see that TallyAge is basically in sync with data from other clinical trials testing epigenetic aging clocks. I also want to share a couple key insights that we've gained about the test beyond users just getting younger over the course of the year. We have found that a variety of things can influence the TallyAge test. So this is just a quick PSA. If you're going to take the test, don't take it when you're sick. We found that things like COVID or a non-COVID respiratory virus can spike TallyAge. So it's critical to take the test when you're feeling healthy and not sick. And in conversations with users and information we've received, it's also becoming clear that modifications to the sample site, the mouth, can influence the results. So as an example of this, being sick is an obvious indicator because the mouth is the first line of defense for the outside world and pathogens. But doing something like routinely drinking or using alcohol-containing mouthwash that sears the inside of the mouth would be expected to influence the results. If something happens that causes vomiting or damage or irritation or injury, these things might also affect the result. So we're actively studying all this and doing research, but I just wanted to share that it's really important when you take the test to make sure that conditions are normal and you feel healthy, and that'll help optimize the results over time. So I want to end on an exciting slide before we jump right into questions, which is that we're always collecting new data and actively doing new research here at Tally. And we've added 4 new questions to the lifestyle survey. Specifically, we now ask about intermittent fasting, about sauna use, about overall oral health, and time in nature, the latter of which is an interesting blend of a mental health and exercise activity. And the next time you take the survey, we'd love for you to answer them and help us gain new insights into what the TallyAge test correlates with. And this kind of data helps us power possible recommendations and other tools. So with that, I think I don't want to take any more time and just jump right into answering as many live questions as possible.
Awesome. Thank you, Adiv. I think we can stop sharing and we'll jump right into questions. I just wanted to let everybody on the call know that this call is being recorded and we will follow up with the member community via email with a link to the replay that will be available. So if you want to reference back, or for some of you who might have joined late, you can watch the beginning part again. Great. So we have a lot of questions in the chat, so I'll just get right to it. The first question that came up actually a couple times is, how has the process of determining epigenetic age changed over time?
Yeah, that's an excellent question. So I think in 2013, when Steve Horvath published his paper in Genome Biology and Hannum et al. Published their paper in Molecular Cell, it was just fascinating that you could even do this, right? Nobody knew that you could predict someone's age based on DNA methylation. And I think in many ways, this was a prelude to learning and having confidence that epigenetic alterations is a really bona fide hallmark of aging. But the shift over time has really been moving away from, okay, how good are we at predicting age, to how good are we at capturing health and outcomes. So we've kind of migrated away from a forensics types application to more of a prognostic aging biomarker type application. And when new tests come out, like the TallyAge test, really the focus should be, how is this relevant to health and wellness? And using DNA methylation sites that change with age and also are reflective of things like overall health and lifestyle. That's really what the major shift has been for the field.
Yeah, and I think that's one of the key differentiators of the TallyAge clock also, is that, when we did the training, not only did we have that large data set and that diversity within the data set, but that you, Adiv, and the rest of the science team were extremely focused on validating the data coming out of the clock against real lifestyle and health factors. Interesting question here around differences between male and female. So has the testing revealed any significant differences in TallyAge or how men or women are aging?
Yeah, that's a really excellent question. So many people are probably aware that there are sex-based differences in aging in humans and in animal models. And males and females have different risk factors for age-related disease and mortality. And when it comes to lifespan, males don't live as long on average as females. And a great indicator of this is that the ratio of female to male centenarians is about 5-1.
Wow.
And that's really quite compelling in terms of illustrating that difference. And we see that on average, males are significantly older than females when it comes to TallyAge. So that difference that we see in statistical data and epidemiological data is also being picked up by our TallyAge test.
Interesting. So a couple of questions about the test itself. Does the TallyAge test identify genetic deficiencies?
The TallyAge test is not a genetic test. We're not actually reading what your base pairs are. We're looking at what the methylation status is of specific sites. So specifically, we want to know what sites are methylated, what sites are not methylated. So it does not provide information about genetic deficiencies. Now, that being said, it is entirely possible that different genetic conditions could influence TallyAge. And we have done an analysis in a publicly available data set in progeria patients. These are patients that have accelerated aging and die very, very young in life. And we see that TallyAge is much higher in those patients. So we do know already that genetics can play a factor.
Customers are asking if they can get the actual mapping of their methylation test results. And I'll just take that one quickly. The answer is yes. We keep everyone's data secure and private, but if you request it from us, we are happy to send you that direct download of information that you can have. Another question here is around the accuracy of the cheek swab test versus a saliva test versus a blood test. Can you comment on that?
Yeah, definitely. So what we did here was pretty innovative. at Tally. I just want to comment that it wasn't clear you could make a next generation clock based on a cheek swab. Now clear that you can. But at the time, before we embarked on this quest, that was a hypothesis that we had to, explore. And one thing we've done to really optimize reliability of the test, as I mentioned before, is we filtered out DNA methylation sites that are less stable in a 24-hour period. Now, we put in a huge amount of effort in terms of research to make that test-retest difference as small as possible. And we actually employed a very novel computational approach to make that happen. We can't share too much right now, but additional data and details will be forthcoming soon in a peer-reviewed document paper. So I think wait a little while, and we can probably give you a lot more details for that one.
Wink, wink, nudge, nudge. More info coming there. One question here is about how the Tally test does not include information that outlines predictive longevity. Why is that?
Predictive longevity. Yeah, so I think this is basically asking, why aren't we telling you how many years you have left to live? Yeah, unfortunately, I don't know that any test in the world can do that. That would also be really creepy if it could. But the way this works is, for any aging biomarker, it's more of a risk factor, right? So let's say you look at a test that says, okay, people that have a higher epigenetic age are more likely to experience this event over time, and you have a longitudinal study. It's not that you're guaranteed to experience that event. It's not that the event is guaranteed to happen at any point in time or any period of time. It's just indicative of an increased risk for that event. And so that's the way to conceptualize the delta age value, is not how many years you have left to live, but as a value that is basically giving you an indication of how you compare to the cohort it was trained on.
Awesome. There's a question here around the methylation markers. So there's a question around how many methylation markers are we actually looking at? We've cited in the past that we generate data on some 850,000 methylation markers, but we generate the TallyAge on a subsegment of that data. So the question is, what do we do with the remaining data?
Yeah, so the TallyAge test uses thousands of DNA methylation sites to make an age prediction. We use many, many thousands to make an age prediction. But we have filtered out most of the methylation information that's collected for two reasons. One, it doesn't correlate with age, or it's too noisy. So this goes back to when we looked at samples over a 24-hour period. Methylation can be a very dynamic process that changes all the time, right? So if I get sick, I need to change my methylation to change the expression of immune genes. And we didn't want to look at methylation sites that were likely to change radically day to day. And so that's why we filtered out the majority of that information, because we found it was actually counterproductive for age prediction.
How do biological age tests of individual organs that can age at different rates compare to Tally's test, and are both needed to get an accurate assessment for individuals?
Yeah, that's a really, really good question. My answer to that would be that each aging biomarker provides a unique insight into biological age, right? So you can think of something like a next generation epigenetic clock like TallyAge as something that shines a window into biological age. But biological age is so complicated that you realistically need multiple windows to get a complete picture. And so I think that doing something like an epigenetic age test that's next generation and grip strength and HbA1c and these other longevity markers all together is a really holistic way to think about testing for longevity. And in terms of organ-specific biomarkers, it really depends on what you're talking about. But I generally think that when it comes to epigenetics, it's more valuable and scientific to get a readout from the tissue you're collecting.
Great. There's some questions specific to results. I'm going to shift a little bit away from the test to talk a little bit more about people's results. One of our members says that they saw a big jump in between tests. And what are the reasons or most logical reasons potentially that stand out about why that might happen?
Yeah, that's an excellent question. We'd have to talk to that person to learn a lot more. And there are many, many explanations for this. I hope this person doesn't mind me sharing, but I talked to someone, for example, just to give you kind of a more outlandish story, that was not happy with the results. And after a discussion, it turned out that they had engaged in a ceremony that caused potentially quite a bit of vomiting, and then took the test shortly afterward. There are definitely things that can transiently affect the results. And so I think anything that causes inflammation of the mouth or prevent irritation of the mouth, I would hypothesize to affect the results. Now, there are other explanations as well, including things that we don't know about, because we see that there are so many holistic lifestyle factors that influence TallyAge. There are probably many more waiting to be discovered. And of course, as we discussed before, even though we optimize the reliability as much as possible, you still can see shifts between tests. And that's why we really emphasize the importance of a trend over time, as opposed to putting all of your stock in one test. And by the way, that's true for any marker at all, is the trend over time is what you're looking for. You don't just want to look at one result and say, I'm good, never again. That's it's a rare thing to happen.
Right. And you did mention COVID earlier. There's another question here around do other viruses like COVID or HSV1, does that potentially increase the rate of aging?
Yeah, so we have seen that both COVID and non-COVID infection significantly elevate TallyAge. And what that also suggests, and by the way, many of the methylation sites we look at are involved they're connected to immune genes and inflammatory genes. So this all suggests that things that impact inflammation in the immune system are likely to impact the TallyAge result.
This is an interesting conceptual question from one of our members. Absent any lifestyle changes, is it correct to assume that we should expect TallyAges to trend up about 1 year over the course of 1 year?
Yeah, so on average, if you look at a group of people, that is what we would expect to see. If truly nothing changes, if everything is the same, which would be impressive if you could even do that. But yes, that would be the expectation.
Another question here around results is that one member's TallyAge dropped about 2 years after the first test. This person is in their 70s, but this person does not feel any better than before. So should this person be concerned, or is this a rather normal fluctuation?
Yeah, well, I'll just say a couple things. That something can improve, but that doesn't necessarily mean that it's obvious or it's clear. I mean, this is a terrible example, but let's say I go from having 2.5 servings of vegetables a day to 3 servings of vegetables a day. I don't know that I'm going to notice a massive difference in my day-to-day, but I do know based on published research that I'm probably going to be healthier and live longer based on that. So I just want to point out that changes are not always incredibly obvious. In terms of fluctuations, most people, if you say, hey, take the test a bunch of times in 1 day, the results are going to be between 1 and 2 years apart. So that differential is typical to see. And there are probably technical and biological reasons for that.
Yeah, I did see a question in the chat about, how it's great that we were able to reduce the noise in the aging clock with some of the proactive steps that we took around the 8,000 samples and the replicates, et cetera. So I think you just also hit the other piece of that, which is that that retest reliability is within that 1 to 2 years, which is it's quite good.
Yeah.
Just shifting gears a little bit, I think this one we actually get quite a bit, this question. How do the answers to my lifestyle quiz influence my TallyAge?
Yeah, well, the lifestyle survey score significantly correlates with TallyAge, which is unsurprising given that the lifestyle survey asks about many of the holistic lifestyle factors that we already saw correlate with the test. And so we are collecting data over time, and we want to explore this more to answer questions like, when someone follows their action plan and improves their lifestyle survey, what is the average reduction in TallyAge? So I think the honest answer there is we still need to do research and really explore this, but I can say on a high level that the lifestyle survey score and TallyAge do significantly correlate with each other.
Great. And just to be clear on that, though, your TallyAge is calculated 100% from the DNA methylation.
Oh, absolutely. Yeah, you don't need to submit a survey at all. The way this works is you send a sample in, our lab partner extracts the DNA methylation information. Those DNA methylation values are plugged into our machine learning model that spits out a DNA methylation age that we call TallyAge. So it's as objective as possible and uses nothing but the sample.
Great. So we have some members who heard your PSA about how to take the test, and we have one member that says that they took the test first thing in the morning, right after waking up. Heard your comment about mouthwash morning and evening after brushing in this person's case could affect the results. So is testing immediately in the morning enough to avoid skewing the test, or is there something more drastic required, something more ideal?
Yeah, we would recommend taking it first thing in the morning before doing anything. And that's certainly how I take it. And just like a primer on mouthwash, if it's alcohol containing, given that alcohol is an incredibly significant factor for TallyAge, we don't know this. We want to investigate it, but we certainly hypothesize that that would have a negative impact on the score. So if you're going to use mouthwash, I would just be aware that alcohol-containing mouthwash might be an issue.
Right. And speaking of alcohol, given that both alcohol use and stress are significantly impactful to aging, one member would like to know, how would you address the paradoxical stress reduction benefit of moderate alcohol use?
That is a really good question. That is a tough one. So it is interesting. A lot of the early research on alcohol intake suggesting that a drink a day was good for you actually included people that don't drink anymore because they were either former chronic drinkers that stopped, or because of, say, health issues that preclude them from drinking. And once you control for that, it's pretty clear, or it's very hard to argue, that any amount of alcohol is good for you. Now, does it relieve stress? 100%. I don't think there's any question about that. And I don't think we're telling people never to drink, just to be aware of the fact that even moderate amounts are probably not optimal. I think that the solution here is to try and manage stress a different way, if possible. I understand that's easier said than done, but that would be the best of both worlds. And if we just look at our own results, we see that stress is not as significant as alcohol. So I don't think they would balance out perfectly.
What is the spread between the biological or the TallyAge that could set off an alarm to somebody? Is there a range where somebody should be nervous or concerned?
Yeah, I think that the vast majority of people are plus or minus 3 years. And then we see that, it gets progressively less common to be further and further away. And I just want to caution people, if you get a result that is higher than you'd like, the solution should not be to panic and, immediately worry, because again, as we discussed, there can be other factors at play here that explain it. And so, I think what is typical is for someone to test again or wait for the next test and see how that pans out in comparison. But it's very difficult, of course, to know, why someone is +7 or +8, which is more rare to see that spread without having a lot more information. But what I would say is, if you get a result like that, don't panic, and wait until your next test to see if it's consistent or if maybe it was due to some other factor.
We have another interesting question here specific to menopause or perimenopause. Have you seen in the data how being in that stage could affect epigenetic markers and/or our TallyAge test results?
Yeah, so that's a really fantastic question. Research from other groups has suggested that menopause can accelerate epigenetic aging, and that finding is interesting in the context of menopause increasing the risk for things like osteoporosis and bone fractures. We, have started to explore this, and we've done some preliminary analyses here at Tally, but I think we probably need to do more research to really get at the heart of that question. But certainly there have been published reports suggesting that that's the case.
Great. We're going to take a little bit of a shift over to supplements. I see a bunch of questions around supplementation. What is the difference between Vitality and Amplify, the two supplements that we offer at Tally, and is it best to be taking both?
Yeah, so Vitality was really inspired by Dr. David Sinclair's supplement stack and contains 5 ingredients: resveratrol, calcium alpha-ketoglutarate, quercetin, fisetin, and spermidine. And these are ingredients that were each included for a very specific reason. And they all target aging hallmarks in different ways. Spermidine, for example, is a very potent promoter of autophagy, which is a cellular recycling process that gets rid of old junk and proteins and really breaks it down and reuses it. It has a very, very positive effect on a cellular level. That's the main goal of Vitality, is to really fundamentally address all 12 aging hallmarks. Now, Amplify, which has 3 ingredients, berberine, glycine, urolithin A, also amplifies that effect. And there's clear mechanistic data that it can target and influence aging hallmarks, but it's really more focused on metabolic health and energy. So, for example, berberine is something that clearly influences sugar metabolism. It also is a potent activator of AMPK, which is a metabolic sensor that gets triggered by things like fasting or prolonged intense exercise. So the way I would frame these is Vitality is more of a foundational supplement for dealing with the hallmarks of aging, which include things like, by the way, macroautophagy, loss of proteostasis, epigenetic alterations, telomere attrition, etc. Whereas Amplify is designed to really further target, but also has an emphasis on metabolism and energy production.
Awesome. And just back to basics, a couple of questions here around taking the supplements themselves. Do you recommend them with food, without food, late at night, early in the morning? What's your take?
Yeah, we definitely recommend taking the supplements with food. And there's actually a lot of research on this, that people just tolerate supplements so much better when you take them with meal. I mean, this is not the case for every single supplement, but it's just by and large a good recommendation to follow. And we have had people report being more energized by these supplements. So that's one reason we would suggest taking it earlier in the day, maybe not right before bed, in case you get hopped up and are too excited. But generally speaking, morning or afternoon would be our recommendation. If you fast, we know many of our users fast, we would say wait until the first meal of the day.
And how important is it to take the supplements on a consistent basis? Meaning for these two in particular, taking them daily. Sometimes our members miss a few days. Is that going to be an issue?
Yeah, that's an excellent question. If we look at the clinical trial data for these ingredients, I mean, you look at, say, trials using berberine for 600 milligrams or urolithin A for 250 milligrams, or any dose. Typically, they give them every single day, and people take them consistently during the trial period. So in terms of reported benefits and published clinical data, what we have to look at is a situation where it was consistent and daily. That being said, life is complicated and messy, and sometimes it's not possible. You're traveling, you forget, and I don't think people need to worry, too much about that.
And what should people look for or look to see potentially in the TallyAge results or otherwise as they're taking the supplements to know if they're working? And is there anything noticeable that might be obvious to them that they could, say, hey, this is reassuring me that these are actually working?
Yeah, everyone is different. So it's going to be very, very personal, and it depends on a lot of different factors. But we can share what users have written in to us. People have written to us and said that they've really felt more energetic. They felt that these supplements have helped address their fatigue. Other people have said that they've been able to exercise harder for longer. Someone else mentioned that they feel that their GI system has generally been, more optimized and stabilized. And so really, I think that's the thing about interventions, is the effects are often very personalized. And I think it's up to each person to kind of decide, what am I noticing here? And by the way, measuring is a great way to be more objective about it. If you have a wearable, if you get, lab tests done, that's another way to objectively look for improvements outside of the subjective experience.
Yeah, and I think that was the theory behind launching our action plans, right? Was to help people to understand the lifestyle interventions that could have the most impact for them specifically based on their DNA methylation profile, so that they could have a place to start. And then having the Tally test as a measurement tool to track progress over time so that you can start to see a reliable trend line. And then certainly the supplementation is an extra boost to help with that. Last question on the supplements before we switch to a fun lightning round. Is what is the current view of Tally scientists on NMN supplementation and its impact on longevity and biological age? Similarly, what about metformin?
Yeah, wow, really good questions. So NMN, nicotinamide mononucleotide, is one of many NAD precursor molecules. Niacin or vitamin B3 is one, but it's less effective. Nicotinamide riboside is another one, which is very similar to NMN. NMN is NR with a phosphate group on it. And these all boost NAD levels. It's just unambiguous that if you take them, you get significant increases in circulating blood NAD. And we know that NAD levels decline with age on average. We know that people that exercise are more likely to maintain their NAD levels as they age. And we have quite a bit of evidence in small mammals, mice and rats, that NAD precursor molecules slow aging or increase health span. So they shift aging trajectories for the better. And there are exciting clinical trials out reporting benefits for these molecules. Nothing is really long-term just yet, but I think it's an exciting field to track, and we're definitely watching it very closely. I'll just mention briefly that there are other paths to boosting NAD. Urolithin A, which is found in Amplify, for example, has been reported to elevate NAD in mice. Now, we don't have any published human clinical trials yet, but there is a study suggesting that urolithin A did a better job than nicotinamide riboside at elevating NAD in mouse skeletal muscle. So there are many paths to Rome, probably, when it comes to NAD boosting.
And metformin?
Metformin. Okay, so metformin is interesting because when you give it to mice, there's no doubt that the mice tend to be healthier and seem to have a better health span. But one thing to keep in mind is that the typical mouse experiment situation honestly sounds kind of awesome, kind of being like at a resort forever. Basically, you're in a cage, you have unlimited access to food. And a lot of mice just kind of lounge around and eat all day. And so I think one thing to keep in mind is that metformin being beneficial in these realistically metabolically unhealthy mice is a different situation than more real world conditions. So is it abundantly clear that metformin is amazing for diabetic patients, pre-diabetic patients, and health outcomes over time? No doubt about it. Is there compelling evidence that for someone who's not pre-diabetic or not diabetic, that metformin will increase longevity? Remains to be determined. I know that Dr. Nir Barzilai is trying to get funding for a clinical trial called TAME to test this in older adults. And that would really be the ultimate test of, is metformin a longevity drug?
Super interesting. Okay, quick lightning round, Adiv.
Okay.
Ready? Okay.
Let's do it.
Steam room or sauna?
Both. So good.
Traditional sauna or infrared sauna?
I'm team traditional sauna, personally.
Cold plunge?
No, I have no objection to the other types.
Cold plunge or no cold plunge?
Definitely cold plunge.
Cannabis?
I look, there's no doubt that it can be very relaxing. There's a lot of people that have reported huge benefits. And there's actually some surprising research going on involving a variety of conditions. So there might be real therapeutic potential. Do I think it's beneficial for aging for most people? Probably not. That's my perspective.
Great. Thank you. All right, we have some interesting questions that I think sort of fit into a research kind of future bucket. We also have somebody who has shared some really nice accomplishments from themselves. So this member says, thank you so much for Tally. When I read Dr. Sinclair's book, Lifespan, I tried to do as many protocols as I could. I lost over 100 pounds and got to where I run about 40 miles a week. I eat once a day and mostly plants. And the new Tally protocols are helpful too, and feel like my life is really improving. Are there any clinical trials that I could join? I'd like to be doing as much as I can to live forever. So first of all, to that member, thank you so much for sharing. We are so happy and proud of what you've accomplished. That is a lot of really fantastic work. So well done to you. And Adiv, are there any additional trials or other things that this member might look into?
Yeah, off the top of my head, one thing you can do is you can always look on clinicaltrials.gov, or you can reach out to associations. Actually, if you send us an email, we're happy to provide some of these details and resources that can help give you specifics. But I also just want to echo, that's really impressive and amazing, and we're really, really happy to hear that you've made so many profound changes in your life.
Yeah, please do follow up with us at hello@tallyhealth.com on that question specifically. But again, any of you, if you have additional questions that maybe weren't covered or follow-ups that as you think about the discussion today, again, that's hello@tallyhealth.com, and we will try to get back to you as soon as we can. Just a few more, we have some time here. Stem cell therapy, relatively new, but very interesting. Can that have an effect on TallyAge?
Yeah, we don't know if it can, but we would love to know. And, we'll keep an eye out for publicly available data that's interventional involving that. Stem cells are very exciting. They have huge therapeutic potential, whether we're talking about induced pluripotent stem cells or mesenchymal stem cells or embryonic stem cells. The research is ongoing, and mostly it's looking at very specific conditions right now, but definitely we'll watch out to see if there's data sets we can analyze to help answer that.
There's a question here around our future research. Are there any specific areas that you, Adiv, or the rest of the science team at Tally find interesting to do additional R&D, whether that be just in general in terms of aging and longevity and how we can improve or reduce our epigenetic age, or additional supplement areas for specific areas of health?
Yeah, well, we can't give too much away, but we are always interested in innovating and coming out with new products to meet health and wellness needs. So for sure, on the supplement front, we are constantly innovating and exploring the literature. In terms of the research front, we really want to continue to understand DNA methylation and aging because it's really complicated. This is why we have to use machine learning to try and mine the data. And part of what we alluded to earlier, that may or may not be coming out soon, it does contain a really robust exploration of the epigenetic patterns in a cheek swab with age and some underlying biology. But that's something we really, really want to better understand. And we'd love to get to a point where we can look at an output and say, this is higher or lower, probably for these reasons, because of these signatures. And that just requires a lot of research to try and do. But that, maybe one day the field will get there.
Awesome. Two sort of more corporate questions here, and we are winding down on time, but I'll just go through a couple more here. Is Tally HSA or FSA available? The answer to that is currently no. However, we are working on it. And I think that the one thing that our member community can do to help us with that is ask about it to your doctor, or ask about it when you're having conversations with your insurance provider. We would love to be able to make this eligible so that we can have more people be able to access our service. Another question corporate wise are, who are your founders and where are you located? So we have two co-founders. One is Dr. David Sinclair, who is one of the world's most well-known longevity experts, and he's been running a lab out of Harvard for the last 30 plus years. Our other co-founder is Whitney Casey. She is a serial entrepreneur and currently is a partner at the private equity firm L. Catterton. So the two of them came together and came up with Tally. And Adiv and I, as well as a number of other employees, were very fortunate to meet them and to be able to turn this into a business. Our team is scattered around the US. We are all remote. We do have a fairly large group of us in the New York, New Jersey area, but otherwise we are kind of all around the US. Okay, last but not least, one more question before we wrap up. I think this is a great one for you, Adiv, and I'm going to read this one verbatim. My friends all jokingly ask, how do I know that it's not a scam, that my tally age started higher than my chronological age and has been going down? They say that's a great way to keep customers coming back. Any advice on how I should respond to them?
That is a very funny question. So that's great. I'm really happy that it's gone down. I think the answer to that is, if you look at the data we presented during the webinar, right? Humans are heterogeneous, right? Not everyone increased their lifestyle score. Not everyone reduced their tally age. So I think the data speaks for itself, that there's a diversity of responses and trajectories that are possible and exist.
Awesome. Well, thank you all so much for joining. We are out of time, but like I said, if your question didn't get answered or if you have additional questions, please do send them to hello@tallyhealth.com. I also want to remind you all that we recently launched our referral program where members like you can refer a friend to join Tally, and you both will get a credit of $50 towards your next membership fee, and that is uncapped, so as many as you want. So your unique referral code is in your login screen. Once you log in, you'll see that. And if you have any questions about that, please, again, feel free. Our team is here to help you with that. But we're super excited about you being able to give that credit to your friends and spreading the word. We really appreciate you being part of the Tally community. Thanks so much for spending time with us today. And we really look forward to seeing you again soon. Take care.